Hello again, it's Dr. B. This is a shorter recording than the last one because a lot of the organelles and cell parts that I covered with prokaryotic organisms, a lot of those serve the same purpose as in the eukaryotic. However, you can even tell by this diagram that eukaryotic cells are more complex. They clearly have a lot more parts, and that's partially why they take a lot longer to reproduce. They're much larger, etc. So I'm going to talk about some of these that are different and or some of these organelle parts that act in a different way or function a little differently than the ones in a prokaryotic cell. So basically the first thing, let's again start from the outside, is we see on the outside that looks like there's some hairy-like things called cilia. Now not all eukaryotic cells have that, but understand one thing, that prokaryotes do not have cilia. Cilia are like little hair-like things that either help to move minerals and food across from one location. They're also used for sensory to some degree. And sometimes they can be used for things to stick onto, but those are not found in bacteria at all. So we see here that the outermost part of this particular animal, and this is an animal cell, there is no cell wall. And if you look around here, you're not going to see cell wall, and that's because there isn't one. And so given this particular thing, you'll see that the plasma membrane right over here is the outermost, and it's very jelly-like. It's not very solid. It's kind of fluid. It's almost like Jell-O. And floating around this like ice cubes in a punch bowl are all kinds of proteins that allow certain materials to go in and out. That's how things do go in and out, typically through some sort of facilitated diffusion. So then, of course, we have the blue area, which is the cytoplasm, and I've already covered that. But I'm going to cover some of the ones that are a little different. And I'm going to start right here. So this big purple blob here is called the nucleus, and that's probably not new for you. And that is where the DNA is stored. And remember that DNA is like gold for Fort Knox. The cell doesn't ever want to let its DNA outside of its nucleus. And so the way that it produces proteins is it makes a photocopy of a gene from that nucleus. And that photocopy is called RNA. And so the DNA is found in here. That's the codes to produce the actual cell and all of its parts. But the photocopy is sent out to these little teeny tiny, the photocopy called RNA is sent out to these little teeny tiny dots that are found on this area called the endoplasmic reticulum. And those dots are called ribosomes. And as you know in the last video, they read the RNA and they make the proteins. And of course they're hanging out at the club called the endoplasmic reticulum. This is the rough endoplasmic reticulum, and it's called the rough one because it's studded with all of these ribosomes. There's also a smooth endoplasmic reticulum, and that is an area over here, and that is basically where a lot of your hormones are made, steroids, things like that. And so they're smooth because they lack ribosomes. So let me go back for a second and now talk about one of the most misunderstood parts, and that's called the nucleolus. I've also heard it pronounced nucleolus. But this is an interesting story because of the idea of what came first, the chicken or the egg, in the sense that if a cell has DNA, makes an RNA copy, and sends it out to the ribosomes, which basically are a little piece of protein that codes them into proteins, the RNA, pieces into proteins, then what made the first ribosomes? Like, how are ribosomes made? And basically that's where they're made. There's other things the nucleolus does, but it's the site of ribosome synthesis, and that's where they're made. And then shuttled out, so that's pretty neat stuff. And of course they leave by the little pores in the nucleus. That's all nice and good. So a couple other major parts. Mitochondria. You've probably learned this as, the powerhouse of the cell, and it's where cell respiration is done, and it's where the cell takes in sugars and oxygen and gives off carbon dioxide and a little bit of water, and it makes ATP, so the cell has energy to do stuff. The part that's interesting is those mitochondria are about the same size as a lot of bacteria, and mitochondria also have their own genome. They have their own DNA. They pretty much replicate when they want. And so there's been an interesting scientific theory that perhaps these mitochondria used to be ancient bacteria that long ago kind of made their way into our cells and just kind of stayed there. And so that's an interesting theory. Couple other words, not many others to talk about. Lysosome is kind of like a little stomach, little intestines of the cell, and it's basically filled with enzymes, and it's the way that the cell can break down particular food sources and waste products. As is this one called peroxisome, which makes sense. This one basically is a way to break down peroxide, which is kind of a a bad chemical for cells. It breaks it down into water and oxygen, which is not. Centrioles it uses only when it's dividing in mitosis, particularly very active during probably anaphase. And microtubules are kind of like the little tubes where the cell can transport different things it makes from different places. The Golgi apparatus, going around here, the Golgi apparatus is basically after the proteins are made. They need to be folded up and packaged and shipped to go other places in the cell or outside of. So they're kind of like the UPS of the cell. That's kind of what they do, is they package and deliver stuff. And based on that, I think that's a really pretty good estimation of most of the other parts that you might need to know. All right.